This is Lecture 9 for Biology 178. Starting the cardiovascular section, we'll be focusing on hematology, or the study of blood, which is why I have a picture of the artist Aaliyah as she was doing her role of Ashanti, the Queen of the Vampires, from the early 2000s movie Queen of the Damned. So, the cardiovascular system itself is a three-part system that consists of a pump, which is the heart, and propels and maintains blood pressure, conducting tubes, which are your blood vessels, so arteries, capillaries, and veins, and then a thing that travels through all of these, which is the fluid, blood. Blood transports a large variety of materials, assists in temperature regulation, and aids against the defense against disease. So the major functions of blood are as follows. It transports dissolved gases, nutrients, hormones, and metabolic waste throughout the body. It's the major transport system throughout the body. It regulates the pH and ion composition of interstitial fluids and can have those get eliminated through the urinary system. It restricts fluid losses at injury sites, so it is able to clot and stop wounds. It defends against pathogens and toxins coming in the body. Does this with the help of white blood cells that are in the blood itself. And finally, it stabilizes the body temperature. So temperature in the body is going to primarily be measured through the blood, and as we cool the blood down, we therefore cool our body in core temperature. This is why you might get flush overall when it's very hot and you sweat. You're trying to cool down the blood to stabilize your body temperature. Blood itself is a fluid connective tissue with a unique composition. Like all connective tissue, it consists of a primary cell, in this case the formed elements and cell fragments, and then it consists of the matrix, which in this case is plasma. Plasma is going to be a watery ground substance that has protein fibers throughout it. Now, as far as amount of human blood in the body, there's about 5 to 6 liters in males and 4 to 5 liters in males. This difference is only due to the average body size, in that males are typically larger than females. Of course, this is going to be the case across all humans, in that larger humans will have more total blood in their body than a smaller human. As far as the composition of whole blood, like I said, it consists of plasma and formed elements. Now this is about half and half. Typically there's going to be a little bit more plasma than there are formed elements. Take note of the range on each of these. The range in plasma is 46 to 63%. The range in formed elements is going to be 37 to 54%. So this is going to vary, and a lot of this can vary depending upon the altitude that you live at. People that live at higher altitude will have more red blood cells. It could be your hydration levels affecting plasma concentration, etc. Plasma itself is composed of plasma proteins and a variety of other solutes, but mostly water. The plasma proteins are albumins, globulins, fibrinogen, and then there's some different enzymes and hormones that are in there. The different solutes that plasma contains are electrolytes, so salts, potassiums, different phosphates, calciums, other organic nutrients and organic waste that need to be processed in the body. Formed elements are composed of platelets, white blood cells, and red blood cells, and we'll talk about those a little bit more. Let's start with plasma. Plasma is actually going to resemble the composition of interstitial fluid in many ways, specifically that it is 92% water. Plasma exists because of the continuous exchange of water ions and small solutes across the capillary walls. So this is why it's going to be the primary fluid medium through which all things travel. The rest of plasma is going to be composed of plasma proteins and other solutes. So the different compositions we've already covered are as follows for plasma: the proteins, solutes, and water. Remember that plasma proteins are things like albumins, globulins, and fibrinogen. Fibrinogen is very important as a clotting element in the plasma, and we'll talk about that more when we talk about hemostasis later in this lecture. The other compounds are going to be used to bind different types of compounds in the body. Globulins, for instance, could bind different hormones as they travel through. The solutes are going to be things like electrolytes, organic waste, and organic nutrients. These would, of course, be things that are just transferred to the body to other cells in order to be processed or to be eliminated. Now that we've talked about plasma, we can focus on the formed elements. Formed elements are going to be composed of red blood cells, white blood cells, and platelets. A majority of formed elements are going to be, of course, red blood cells, also known of as erythrocytes. These are going to be responsible for gas transport as they transport both oxygen and CO2 in the body. White blood cells are otherwise known as leukocytes. They are fully functional cells, so they have a nucleus, they have organelles, etc. And they are generally involved in the body's immune system. There will be a separate bonus lecture in addition to this lecture provided that is going to cover in depth the white blood cells. Finally, platelets are otherwise known of as thrombocytes. These lack a nucleus, and they're not really a cell at all anymore. They're more cellular fragments. These are going to be like bricks that exist in the blood and are responsible for coagulation. Formed elements are all initially formed in the red bone marrow, which are going to be found in different long bones throughout the body. These are all formed initially from hematopoietic stem cells, HSCs. HSCs are self-renewing, multipotent stem cells. Because they're multipotent, this means that they can give rise to more than one type of cell. All types of cells, platelets, red blood cells, and all types of white blood cells initially come from this stem cell. Hematopoietic stem cells will diverge into either lymphoid stem cells, which will eventually make a type of white blood cell called lymphocytes. Those are involved in the body's immune system, or they will make myeloid stem cells. Myeloid stem cells create every other type of blood cell or formed element in the body. Now, like I said, there are those two types of stem cells: lymphoid stem cells, responsible for the production of lymphocytes, and myeloid stem cells. What determines whether the hematopoietic stem cell becomes a lymphoid stem cell or a myeloid stem cell is largely dependent upon a signaling agent called a cytokine. This signaling agent can also give rise to a myeloid stem cell becoming a red blood cell, a white, any one of the major types of white blood cells, or platelets, thrombocytes. When we are creating new blood cells, we call this process hematopoiesis, which is the creation or growth of a stem cell into one of the different types of blood cells in the body. What we use to make these cells commit to becoming one of those types of cells is a cytokine. A cytokine is a special type of chemical released in the body in order to signal this cell to grow into that desired cell. In this case, there are three major ones that we'll talk about. The first is colony stimulating factor. Colony stimulating factor is a hormone that is released by activated lymphocytes in other cells during an immune response. So when you get sick or you have some sort of incoming infection, your body will release the hormone colony stimulating factor. This is going to stimulate myeloid stem cells to produce more leukocytes. So if your body wants to get more soldiers to fight off incoming invaders, it signals them to do this by releasing colony stimulating factor. If you start to bleed more often or you're hemorrhaging blood and you need to increase clotting, then your body will make thrombopoietin. This is going to be, or TPO. This is a cytokine that stimulates myeloid cells to produce thrombocytes. So it's going to increase thrombocyte production at the liver. Again, this is made and initiated from, this hormone is going to be initiated from based upon whether or not you're hemorrhaging blood or need more clotting agents. The final type of cytokine is going to be erythropoietin, or EPO. Erythropoietin is a cytokine that stimulates stem cells to produce erythrocytes, more red blood cells. The signaling agent for EPO production from the kidneys is low blood oxygen, or hypoxia. Hypoxia is typically obtained when you go up in altitude or you live there for prolonged periods of time. This is because there's less overall oxygen in the atmosphere up there. So you'll have a harder time breathing, so your body needs to increase the amount of red blood cells to be able to transport more oxygen at a time. EPO is also an external agent that people can inject as a performance-enhancing drug, primarily for endurance sports. So platelet production is called thrombopoiesis. This is going to be where platelets are made based upon a megakaryocyte from the release of thrombopoietin. Here, the thrombopoietin would actually activate on the myeloid stem cell, causing it to become a megakaryocyte, and then it would essentially burst into platelets. These are going to be very important in the function of clotting blood. They are continually replaced throughout the body, and we always keep some production of platelets in the body at any given time, but we increase this production if we're suffering from an injury. The creation of red blood cells is erythropoiesis, and the red blood cell itself is a biconcave disc that has a thin central region with a thicker margin. They lack nuclei at all, they're anucleate, and they lack most organelles, including ribosomes. So they cannot divide or repair themselves, and because of this, they have a lifespan of less than 120 days. Now, red blood cells are formed through the process of erythropoiesis, and this occurs in red bone marrow, which is called myeloid tissue. This is located in the different long bones or proximal limb bones of the body: vertebrae, ribs, sternum, skull, scapula, and pelvis. Fatty yellow bone marrow can convert to red bone marrow in cases of severe or sustained blood loss. This can help to increase the rate of red blood cell formation. This is the process of erythropoiesis. So the multipotent stem cell you can look up as just being the myeloid stem cell. That's what we're actually going for here. So it's going to convert based upon the presence of erythropoietin, EPO. Once that is present, it will convert that stem cell to a proerythroblast. During a proerythroblast, the red blood cell, or in this case, the multipotent stem cell that is converting to a red blood cell, will start to produce hemoglobin. Hemoglobin is an iron-containing protein in the body that carries gases. From the proerythroblast, it will then begin the process of ejecting the nucleus. This is how you know you've reached erythroblast, is the nucleus is shifting to the side and getting ready to push itself outside of the cell. From there, once it removes the nucleus, it will then start to move its way into the bloodstream through the different capillaries that are present in the red bone marrow. Here, it will become a reticulocyte. This is where it enters the blood, and once it enters the blood, it will start to remove the other organelles, the mitochondria, the ribosomes, the endoplasmic reticulum, etc. It's ejecting all of those from the cell. From there, the cell will flatten slightly in the center because it's missing all the organelles, and it will become the fully mature erythrocyte, now capable of transporting gases throughout the body. Here we can see a finished mature red blood cell, and it is composed almost entirely of hemoglobin, hemoglobin being that iron-containing protein that is important in transporting both oxygen and CO2 throughout the body. There are about 1% of circulating red blood cells that are replaced each day in the body, and about 3 million new red blood cells enter the circulation each second. So this is very fast production that's consistent with the body. This rapid replacement rate is necessary due to the relatively short lifespan of red blood cells of just 120 days. Once these red blood cells are done or dead, they actually make their way either to the spleen, liver, or they end up in the bone marrow, and they are engulfed by macrophages, kind of eaten back up, and they're broken back down into their constituent parts and reprocessed. Hemoglobin, remember, is going to be the main composition of red blood cells. The primary function of red blood cells is the transport of respiratory gases, and the molecule responsible for it is hemoglobin. It's abbreviated HB or sometimes HGB. Each red blood cell contains about 280 million hemoglobin molecules, and each hemoglobin molecule contains 4 heme units. So this means that each red blood cell can carry more than 1 billion molecules of oxygen. So this is a rapid rate of oxygen exchange, and this is a highly efficient mechanism for carrying oxygen. About 98% of oxygen carried by the blood is bound to hemoglobin. The other 2% or so is dissolved in plasma. This is going to be a sample of that hemoglobin as it's broken down, and we can see the hemoglobin molecule with the 4 heme units. So it has the globulin protein and the quantitary structure, and then it's going to have the heme unit, where at the center will be an iron ion. These heme units are what allow us to bind oxygen or CO2 directly to a cell. Now, when we actually need to get rid of them, remember I said at the end of the 120-day life cycle, it's going to be transported either to the spleen, the liver, or the red bone marrow, where macrophages are going to break it down. It does this through a process called phagocytosis and lysis, where it's eating it apart and separating the constituent parts. As it breaks down the cell, the red blood cell, the macrophage breaks it down, it's going to pull the hemoglobin out. Then hemoglobin is going to be separated out in the cells into globin and heme. The globin is going to be broken down to amino acids and then just returned to the body. These are effectively proteins that are going to be broken down to their constituent parts. And your body is protein based, so it can use it for a variety of things. The other part is the heme. Heme is going to have the iron ion pulled out of it and recycled to make more heme units later. The other part of heme is going to be the different nitrogenous compounds that surround it, and that will be broken down into bilirubin. Bilirubin will then be excreted, usually in the form of urine, but sometimes it can be processed and put into work as bile in the gallbladder. The reason that your urine is yellow is actually due to bilirubin. This is going to give a slight yellowish tint to urine at different concentrations. Obviously, when it's more concentrated urine, it'll be a darker yellow, but this is what gives urine its color. Now, if bilirubin can't be gotten rid of either through urine, through fecal matter, via bile, it actually builds up in the bodies and then sometimes in fatal levels, and this is a condition that is called jaundice, where the body can't process the bilirubin, and so it gets a yellow tint to the skin. Over time, this can flood out the liver and the kidneys and cause organ failure. I'm sure we've all bled at some point or another. This bleeding is damage to a blood vessel where a blood vessel ruptures. And then we have to get the bleeding to stop. This process is called hemostasis. It is the process responsible for stopping blood loss through walls of damaged blood vessels and establishes a framework for tissue repair. It is divided into 3 phases, but understand that these phases are all occurring at relatively the same time, and they have a complicated cascade of responses that are going to occur. The first phase is going to be the vascular phase. The vascular phase of hemostasis is going to last about 30 minutes after injury and happens when the wound exposes the underlying basement membrane to the bloodstream. Here, endothelial cells will release chemical factors that stimulate smooth muscle and promote vascular spasms. So the damaged cells are going to actually release chemical factors to increase contraction of the blood vessel. It will also have the effect of increasing the turnover rate of cells. So it's going to produce more smooth muscle cells and more fibroblasts to accelerate the repair process. So vascular phase, blood vessels constrict. They release chemicals that make them constrict harder and for longer and increase the rate of cell repair. Here we can see the blades cutting that basement membrane, and vascular spasm happens as the blood vessel tries to close off. The next phase is the platelet phase. The platelet phase begins with attachments of platelets to the basement membrane. Remember that once the wound is actually exposed, that basement membrane is open. It's going to go through the vascular spasm, and it's also going to be sending out a bunch of signals. One of these signals that it sends is going to get platelets to adhere to the area. Once a platelet actually starts sticking to the area, it will send out platelet activating factor. This is going to be released by the platelets to get more platelets to come and stick to it. It will additionally stimulate production of thromboxane, which enhances and continues the vascular spasm. So during the platelet phase, it's also trying to enhance the vascular spasm. Finally, there's going to be platelet factors, which are going to play a role in clotting. They're proteins that are going to come down and help, and they're going to promote vessel repair with platelet-derived growth factor. This general process of getting all these platelets coming down is going to form a temporary platelet plug. Here we can see the platelets that will start to fuse to that basement membrane as they keep trying to aggregate next to each other. So stick next to each other and release different signals to bring more platelets to that area. So the different things that the platelet phase will do will be to enhance the vascular spasm, to get more platelets to stick to the area, to increase platelet production, and to increase the amount of growth and repair that is occurring at the area. The final phase is the coagulation phase. This typically starts about 30 seconds or more after the damage, so it can occur relatively fast. Coagulation is otherwise known as blood clotting. It is a thickening of the blood in general, but this involves a complex sequence of steps that leads to the conversion of the protein fibrinogen, which is just circulating throughout the plasma, to insoluble fibrin. The fibrin is going to combine with blood cells and platelets to create this network and clot the entire area. The events that begin the cascade reaction that causes coagulation are actually different local factors that are going to be chemicals that are released in a sequence of events, starting from factor 1 and working their way all up to factor 10. Once it works its way up to factor 10, this is going to then move through the common pathway. The common pathway is taking factor X, or 10, and using this to turn fibrinogen into fibrin. So here we can see the extrinsic and intrinsic pathways as well as the common pathway. I don't need you to know the extrinsic or the intrinsic pathways. I need you to focus on the common pathway, so the big blue arrow in the center. Our goal is to have the body produce factor X. So once we actually have tissue damage that occurs, a cascade of responses will go on in order to produce factor X. Factor X is a prothrombin activator. This is going to turn already present prothrombin from the platelets into thrombin. Thrombin is thereby going to convert fibrinogen into fibrin. Also in the process and presence of thrombin, thrombin is going to make factor 13, which we'll talk about a little bit later. But fibrin is going to be the end result of that common pathway that will interlink the platelets together, kind of acts as cement in between bricks, sticking everything together. Remember that these clots are only temporary structures. As the vessel walls repair itself following this three-stage process, the fibrin in the clot is broken down and the fragments are removed. Remember that this is going to be a type of positive feedback loop in the body, and that to stop this positive feedback loop, that we actually need to have an inhibition of platelet adhesion in the healthy endothelial cells. So those same healthy endothelial cells release chemicals called anticoagulants. So once the cells of the actual membrane of the vessel are healthy enough, they're going to release anticoagulants to actually stop the process of hemostasis. One of the most powerful ones of these anticoagulants is a protein called heparin. That's going to finish the hematology lecture. Next, we're going to move on to cardiovascular anatomy.